Thank you, Anirudh, for the introduction. Hello, everybody. Welcome to BioNOW Week 4, and we are going to learn more exciting things as we proceed. But today's topic would be Introduction to Pharmacogenomics, and we'll discuss further. I'm not imparting any highly skilled knowledge on you. Rather, it's a learning experience for all of us, and we are learning it together. So let's explore the world of genomics. So the things that we'll be discussing today would be the history of pharmacogenomics, the difference between pharmacogenetics and pharmacogenomics. There will be different types of genetic variations, the drug response, and as we move forward, while we dive deeper into the subject, we'll have a few more webinars on pharmacogenomics, which would include the challenges, the latest research, statistics, regulatory and industry perspective. We'll dive more into the case studies and the clinical trials that's going on, and the future prospects of pharmacogenomics. So let's start with the introduction of what pharmacogenomic is, for better understanding, because usually the field of genomic is considered as a really technical and really scientific field, but what I believe is genomic is a field where it's a window to an individual's past, present, and the future. So we must understand why genomics is really important for us in drug development, whether it is providing the care, whether it is providing the clinical inputs. So it's really important to understand the genetic profile of an individual. So coming back to the topic, what pharmacogenomics is, if we break down the pharmacon, that's the drug, and the genomics is, we know it's a study of an entire set of genes, which is called as genome. So, and genome is really a powerful tiny little thing which contains a lot of information that we are not really aware of, and it provides greater insights in human health perspective and how the aging will proceed. It also provides information about the ancestry and stuff. So it's really important for us to understand what genomic is, what genome profile is, and then when we combine it with pharmacology, when we include the drugs in it, it becomes pharmacogenomics. So when we talk about pharmacology, we have the two terms that comes to mind, there is pharmacodynamics and pharmacokinetics. When we talk about pharmacodynamics, is in some simpler terms, it would be what drug does to the body. It's about the mechanism of action of a drug that happens and that goes in the body. And then when we have pharmacokinetic, it talks about the what body does to the drug, the journey of a drug when ingested, after being prescribed. So what happens is it is absorbed, it is distributed, it is metabolized, and then later on it is excreted. So that's called pharmacokinetics, which we call ADME. Now when we look into pharmacogenomics, it's a study of knowing how the genetic makeup of an individual can affect the way they would respond to a drug, or how the drug would be traveling through the body, and not in the terms of the passage, but in the terms of how it is being metabolized, how it is being distributed, and how it is being absorbed, right? So that's the introduction. When we look at the historical evidence of pharmacogenomics, is it dates back in the olden Greece, where Pythagoras used to present the Pythagoras theorem, right? So he was able to identify the link between the fava beans and hemolytic anemia. along with the oxidative stress. So it's not that it's a very new concept, but again, why it is new and why it is recent is because a lot of evidence was missing in between Pythagoras and Ronak. So so a lot of data that was required to reach to a particular conclusion was missing. So as the field expanded during 1950s, one of the greatest scientists, Frederick Vogel from Germany, he termed this field as pharmacogenetic field to better understand the correlation between the drug and the genetic makeup. Right, and there are a few other significant scientists who contributed greatly. Just to name a few would be Arno Motulsky and Dr. Werner Kellogg. They had really great contribution towards pharmacogenomics and genetics. Now let's understand what pharmacogenetics is and why it's an introduction to pharmacogenomics. So while we are talking about genetics, it focuses on one individual drug-gene interaction. So what's more important is if we study the entire profile, or if we take a complex approach for the entire genome and certain medication, what happens is we get a clearer picture. We get more information rather than going for one gene and one drug. What I would suggest, or what I think, is if we have the entire picture of the whole genome, and let's say if we have got 100 drugs, and if we've got the whole genome in front of us, it will be really easy for a physician or a clinician to determine which drug is more suitable for this individual. So it helps greatly in tailoring the treatment. Again, when we look at the effects, how the dynamics of this whole genome thing works is, if let's say there's a obese person which you presume that would be eating more, and there's somebody who is really skinny, we would think that this person might not be eating more, because we are correlating it with many other things, because food ingestion and digestion and the habits and sedentary lifestyle and so many other things. But what's more important is the metabolism of the food product, the metabolism that goes in the human body. It really impacts your life, and as you can see on the chart, that somebody who is really having fat deposits or looks really healthy would be consuming less, as compared to somebody who is really skinny and active would be consuming more. So that was the example that I gave during one of the lectures, that I do not get satisfied until I complete one large pizza. However, somebody else might just satisfy their hunger with one slice of pizza. So it all depends on the metabolism. While we are talking about metabolism, what's more important is to understand how drug is metabolized in the system, how it is absorbed, and how genomic profile would be creating an impact. So right in front of the screen, what we have is the plasma drug concentration chart, which would indicate the amount of drug that's required to create effective effect, not effective, but therapeutic effect. So we have minimum safe concentration on the top, and then we have minimum effective concentration. Anything below minimum effective concentration will not be producing therapeutic effect on your body. Now, and when we have the maximum effective concentration, or we can call it minimum safe concentration, so that provides a therapeutic window. It's a therapeutic range. So if a drug is absorbed in the system, and if it stays between these two markers, if they stay in between these two, minimum effective concentration and maximum effective concentration, you'll get the therapeutic response. But if it goes above minimum safe concentration, what would we have? We'll have toxicity. The drug toxicity creates serious adverse events. It causes different kind of events, medically significant, not significant. Irrespective of that, any drug concentration that's above that minimum safe concentration is toxic for the body. So how the people would respond differently on this is the information lies in their genomic profile, right? Now these are the genetic variations and the drug responses. So if we have a genetic variation type 1, I'm just giving you an example of how different variants would cause the different type of metabolism. This is a picture of microscopic picture of how any variant or any subtype would create an impact. So if you have a genetic variant, let's say 1, you might respond well and you might metabolize the drug efficiently. However, person X might not be able to metabolize it because the variant that's present in their system is really different, and they might not be able to absorb. So what's more important is Tylenol 500 mg might not be as efficient as your brother or your sister or somebody else who is taking it. So that's the whole point of pharmacogenomics, is to explore how a body would respond to the drug based on their genetic profile. And as we dive deeper into this is just one example of how it's a breakdown of vitamin K using the weak or C1. The body uses it to break down in the system. So there are different types of genetic variations where we have single nucleotide polymorphisms, we have copy number variations, we have indels, we have inversions. All of these, they would create different type of effects, let's say, and you might not be able to respond to the drug in the same way. And so why was the need to understand this correlation? Why was it necessary for the scientist or for individuals who are involved in this study, in this field? Why was it important? So most significantly, one size doesn't fit all. That's the general tagline that I roll with every time, because it is very apparent that if I have a shoe of 8.5, it's not going to fit to you or to anybody. And what's happening right now in the current scenario, Tylenol 500 mg, I'm just giving you an example for better understanding. Tylenol 500 mg is just an example of a drug, how it would react. So what's more important is your body is not meant to either tolerate or to respond to 500 mg of that. You might get a response at, let's say, 150 mg. So why are you getting 500 mg? Now when we spoke about the minimum safe concentration. So this is what happens. The rest of the drug, it goes into toxicity. Although Tylenol acetaminophen has got really good safety profile, apart from Stevens-Johnson syndrome in cases of overdose. But that's one safer drug I'm talking about. Let's say you are taking warfarin. That's the best example because it might cause bleeding, internal bleeding, and we might not know it until it's too late. So what's more important is what if the warfarin 5 mg is too much for you? What if 5 mg of warfarin is not doing anything for you? Right? So you're putting yourself at risk without knowing what drug is going to create an impact on your health. So what are the benefits? If we look at the benefits of pharmacogenomic profiling, is it provides you a personalized treatment. And when it comes to personalized treatment, I've already spoken about warfarin and other drugs. It really helps to understand whether it's a clinician, because self-prescription is something that I don't recommend. So don't go out and say, oh, Ronak told me not to take 5 mg of warfarin. But it really provides more information to a clinician who is prescribing, to say that, look, this person's profile says he would be more tolerant or more respondent to XYZ drug at whatever concentration it is, whatever strength it is, then he'll be able to make an informed choice. So I definitely recommend people, whoever I talk to on this topic, I definitely recommend them to go for genetic testing, get their whole genome sequenced, and get their pharmacogenomic profile done, so that they can better understand what drugs to take and what not to take. Second benefit, if we're talking about the drug safety and efficacy, again, we spoke about serious adverse events and how the effectiveness of a drug is determined. And as we spoke, reduced trial and error of a drug will significantly improve your overall health. It will eliminate any toxicity within your body, and it will eventually lead to better therapeutic outcomes, right? So the doctor's goal is always to treat a patient, make them healthy again. All these pharmaceutical companies, they are relentlessly discovering new molecules and working throughout their lives to provide better healthcare. And in this way, if we have pharmacogenomic profile right in front of us, we are enabling them to succeed in providing the best care possible, right? But again, when we talk about the benefits, it comes with a lot of challenges, right? And there will be ethical consideration. So first major challenge is the genetic complexity. Why the complexity? Because the amount of data that's stored in one single DNA is significant, is more space acquiring than your photographs collectively. Just imagine that. So the complexity is a challenge to tackle on how to interpret the data, how to get things in line with respect to the pharmacological response. We've got a lot of advancement in technology, and with artificial intelligence kicking in and being adopted globally, what's happening is all this data, and whether it is aggregation of the data or whether it is distribution of data, has become a lot easier. The second challenge is limited evidence, is because, as we spoke, since Pythagoras to Ronak, till Ronak, there was a lot of gap in between, because the evidence of pharmacogenomic profile was missing. And there were significant studies that were conducted since 20th century, since the beginning of 20th century. But as I said, it requires highly equipped, high-tech equipment, and the gathering of evidence is really crucial to determine something that's beneficial in this pharmacogenomic profile. The third challenge is data interpretation. As I mentioned, a significant amount of data that's being collected from whole genome sequencing. Again, it requires a highly skilled person to interpret the data, to say this particular biomarker or this particular variant will be showing these symptoms or will be responding well to a drug, and so many other things that's involved in this. So data interpretation is another challenge. And general awareness is, like we said, if people were already aware of things, I wouldn't be sitting here and I wouldn't be imparting the webinar series, because what's more important is for people to understand how genomic data is going to help them, and how they can improve their lifestyle, how they can live longer, how they can eat better, how they can sleep better. All those things, every information is coded within you. So you don't have to look for answers outside. all I can ask is look within you. Again, that's very philosophical of me. And the last one is ethical concern. Of course, when you have such huge amount of data, and when it comes to genetic data, it's more of a fingerprint to you, and we definitely want to secure it. We don't want it to go to somebody else who can make money out of it, who can sell your data on black market to extract benefits from it. And a lot of companies have been doing it. The best thing about BioNOW is that the data privacy is like spot on, so you don't have to worry about it. So again, yes, it is ethical concern comes with data privacy and the consent and so many other things. How a pharmacogenomic testing would be done is the data is analyzed from your blood. We have the sequencers in place that would interpret they would generate the report, and then it will be sent out for analysis, and the final report would be produced. And the final pharmacogenomic profile, not only genomic profile but the pharmacogenomic profile, which would list down all the drugs that are helpful, all the actionable pharmacogenomics will be mentioned on the report, that this person or this individual is really susceptible to taking whatever drug it is, or might respond well to this drug. So all this information will be derived from that, and it's going to be really helpful. And there will be, as I said, personalized medication recommendations are really important, because once you have that information, the clinicians would be in better place to make informed decision based on your profile. Now, when we speak about clinical research application of pharmacogenomics, is first thing we need to understand, as we spoke about the evidence that was missing since these many years. Now we have things in place. Now we have things in place to facilitate this pharmacogenomics, and as you can see on the screen, we have PGRN, we have PharmGKB, we have CPIC, and we have ClinGen, and there are so many other databases that's available and who are actively involved in providing the details on genomics, combining with the pharmacology, so that's drugs. While we are talking about clinical research, a lot of clinical trials are being conducted, but I want to talk about how the pharmacogenomic profiling would leverage or would be advantageous to the clinical researcher is from phase 0, when we are trying to microdose, when we are still trying to understand the basic pharmacokinetics of a new drug or a new molecule. It will be really helpful to correlate that this particular variant profile reacted differently, or this population reacted differently, or if the trials are conducted across the globe, what information comes out from there is we have different demography, we have different races, we have different ethnics groups who would be consuming the drug, and then we'll have more information on how the drug is going to respond on different people, right? So, and again, on phase 1 we're talking about it helps with active metabolites and to determine what type of toxicity would a new drug create on an individual, or whether it is healthy volunteer, whether it is a patient. Definitely we can get a lot of information from this profiling. These are few examples that I've taken on the recent clinical trials that has been going around. There are a lot of categories where the drugs are being observed, and based on the pharmacogenomic profile. Again, it is really helpful to understand what type of response a drug would be creating, or a gene would be producing towards a drug. And yeah, so this was the last slide. I had one more thing to add to clinical research is this is not only limited to clinical research applications. There's a lot of more potential that comes after genomic profiling is whether it is a new drug discovery, whether it is your practice, whether it is your knowing how your day-to-day drugs that you consume, OTC drugs that you consume, would create an impact on your life. So I thank everybody. Let us join hands together to bring healthier tomorrow. And I really appreciate your time. I'm not really sure if I ended too soon or it was too late, but I'll be taking any questions. And of course, we'll be talking more. And this is just an introduction because as we spoke about the general awareness, people are really not aware of this aspect of genomics. In fact, people are struggling to understand how genomics are going to help change the world as we see it. But I'm really optimistic in believing that one day we'll be able to change, create an impact with the genomic information. So thank you so much, everybody. And if you have any questions, I'll be taking them now. Thank you so much.
Thank you so much, Ronak. We're grateful for you sharing your wealth of knowledge with all of us. As for the audience, if you'd like to ask any questions or request to unmute if you'd like to use your microphone.
Well, I don't see. Thank you, Dr. Neda. So is it because I
Mhm.
wasn't very clear on this? Because I didn't explain it well,
That shouldn't be
or
the case. It's an introduction to
yeah.
pharmacogenomics, so maybe people are just like getting into it, getting to learn it.
All right. So if there are no questions, I think it's time to end this.
All right. We'll just give a few more seconds if anybody would want to ask a question.
Thank you, Dr. Minil. Thank you, Maria. Thank you, Patrick. It's all because of you. You you gave me the rub before the game. Thank you, Raju. Yes, we'll dive deeper into more details because we have experts. We have team of experts on our end who have mastered the genomic side of it. So we'll come up with more insights on specifications of pharmacogenomics, or if it is a particular biomarker, or if there's a new study regarding a drug or category of diseases. We'll definitely explore more into that. All right. I thank everybody for attending. And over to you, Anirudh.
All right. So that brings us to the end of this webinar. I hope you've all enjoyed learning about pharmacogenomics. Thank you all for joining in. I would also like to take a moment to express our gratitude. Thank you, Ronak, for taking the time out and generously sharing your experience and knowledge with all of us today. I encourage you all to stay curious, keep learning, and join us next week for the webinar on what is IBS or irritable bowel syndrome by Dr. Krishnan Turbide. Please remember, the more we know, the better equipped we are to make informed decisions and improve our lives and of the people around us. Thank you once again for being a part of this webinar. Goodbye and see you next week.